All right, everyone. So welcome on behalf of all of Aligned to Innovate and the Protein Engineering Committee. I am Dana Courtaud. I am one of the technical project managers helping to organize the tournament. And we're really excited to have this webinar with all of the teams and everyone present to be able to talk about experiences throughout this tournament that's been going on over the last year. So first of all, who is Aligned to Innovate? We are a nonprofit that has a mission to be able to kickstart the use of programmable and automated experiments in biology to help move biology into an engineering-first type of discipline. And we do that by experimenting with different programs that help remove barriers in the field of biology to move forward in this space, working with both scientists from academia and in industry to make really large collaborative projects that are usually quite data intensive and are all available to the public. We accelerate these projects by bringing people together like we are today to talk about goals of the field and have instances for agenda setting. We also facilitate collaborations between different people, different types of science and different countries by having these large overarching collaborations throughout the world. And then, like I mentioned, we remove bottlenecks in various ways by supporting and funding and having project managers like myself help facilitate projects. In this, which is the protein engineering tournament, in this project, we've been able to have a really great group of people who are, I think, all here today, be able to come up with the idea and the concept of this tournament in terms of how the structure of it goes, what the challenge problems were, and how the evaluation metrics and analysis have all been able to come together. It's been quite a lot of work to put this together, and we will be having a paper that will be coming out soon. We have our white paper up on online for people to read through, but we'll be publishing that soon. And this is the team here that made that work all possible. So we have a mix of people from industry and academia and different levels in their career. So it's been really great to get this kind of perspective as we move through and decide what the next steps for protein engineering in the field could be. So if you're interested in future iterations of becoming a part of this committee, please feel free to reach out to us, and you'll see some links to be able to contact us later on. So the tournament's purpose was, like I said, to kind of accelerate the development of computational methods for protein modeling and design. And the concept for doing this is to be able to make this computational engine in the end where we're able to not only predict properties from a protein sequence, but then also be able to have this master goal of then taking properties that are desired and generating a sequence that gets us exactly what we'd like. And to get from where we are today to that end goal, we have created venues for benchmarking, so people who are working in these areas to be able to compete like this tournament, generating open data sets that people can use to make models better and to compare models, and then also releasing automated protocols to the committee or to the community so that these types of experiments can be more reproducible and can expand out and reused, making living data sets years after years. So this tournament that the committee had created and we've done throughout the last year had 2 different rounds to be able to make this computational engine that can do prediction and generation. This allowed lots of different teams to be able to hopefully interact with the data sets and be able to appreciate both having their computational side and then taking their models from computational side and using them to be able to make these generated sequences. And the way that we did that was by having an in silico round that had both a zero-shot learning track and a supervised learning track to make people that had different kinds of approaches be able to use their approaches to evaluate a lot of different data sets. You'll see from the event types that the pilot tournament this year was under the theme of enzyme design. So we had a lot of different data sets for these 2 tracks. And then teams were able to compete in any of the events that they wanted to. This pilot tournament, we didn't restrict anybody from only doing all events or both tracks. We let people pick and choose what they were able to spend their time on. And then they submitted all of their predictions and we evaluated them, and the teams that performed well in at least one of these events were invited then to move into an in vitro round. And this round is where we asked teams to be able to generate new sequences based off of some desired properties that we had. And then we actually, with our partners, were able to make these sequences into a reality and express them and characterize them to be able to choose the winner by experimentation. And the in vitro round is also interesting because we also opened up an area where teams that had a generative approach were invited to compete as well. And so you can learn more about our concepts and strategies for inviting lots of different types of modeling into this same agenda setting venue through our white paper. So please feel free to read more about that. But I just wanted to take a quick moment before we dive into the panel discussion to thank our dataset donors. Our goal in the future is to be able to actively select and create datasets that can be used both in the in silico and in vitro round to build upon them and make them living. But this year in our pilot tournament, we relied on our partners to donate datasets. So we had all of these datasets you see here on the screen, and they were donated from a mix of academic and industry groups. And then our experimental partners were IFF's R&D team in Leiden in the Netherlands. So thank you to them and all of them that are here watching the webinar today. The in vitro round is only possible through your effort. So thank you so much. And thank you to all of our dataset donors. And just so everybody knows, once again, all of the results and analysis that were performed throughout the competition, including these data sets, their problem challenges, and the abstracts of the team's methods are all available on our GitHub that you can find by going to our website, which I'll share a link for at the end of the webinar as well. So I'm going to stop my screen share there. And I think that with that, we can jump in, Chase. Actually, I think I go first, just doing a small intro. Just to have context for the people who are watching the webinar right now, I'm going to do similar to how we had done before, a little popcorn. So for each team, if you could just quickly introduce yourself and the teammates that are here that maybe aren't represented on team or on the screen, if you could introduce yourselves and then just give a very quick overview of maybe like the methods that you used in the tournament. So I'm just going to start at the top and go around, and I'll call you one by one. But the first person on my screen is Michael. Would you go ahead and introduce yourself?
Yeah, sure. Hi, thanks. So I'm Michael. I participated in the tournament together with Robert, who actually did the majority of work, I want to be honest here. But he just became father, and this is why I'm now here instead of him. So, but all the kudos should go to him mostly. I was rather in the background. But I did just a brief background also myself. I did my PhD at the Technical University of Munich in the Rust lab, working mostly on protein language models, figuring out how we can transfer these concepts from natural language processing to proteins. Back then, we established mostly how we can use them for predictive purposes, so assessing the quality of the resulting protein language models on prediction tasks. And now we are looking more towards how we can leverage them for protein design. And currently I'm employed as a machine learning researcher at Sanofi, but the tournament I still participated as a postdoc in the Rust lab.
Great. The next on the screen is Ingmar.
Yeah, hi. I'm Ingmar. I'm the CEO of enzyme, and in the beginning of the tournament, I actually was heavily loaded with coding for this, but then in the second half, Harriet took over, who isn't here because he also recently became a father of twins in his case, so he has two hands full right now. At enzyme, all we do is protein design, and the models that we developed are mostly regression-based models, very little language modeling going on there. We used very basic models in the in silico round, but also later on for the suggestion. Actually, we experimented a bit with new types of models that we hadn't used before. Some models that sample from energy landscapes that Harry just made up on the spot. So we learned a lot in the tournament, and we're really happy about that.
Great, thank you. Next is Jason.
Cool. Hi, everyone. I'm Jason Klima. I'm currently a senior computational chemist at Encodia, Inc. in San Diego, California. And yeah, I'm Team Nimbus from the tournament, the pilot tournament. And yeah, I was I guess a solo team for this round this year. And yeah, the approach I took was a structural modeling approach on all the basically yeah, the enzyme, the pre-transition states of the enzyme protein complexes, and then you scored them with a sort of a myriad of metrics in using PyRosetta, and fit those metrics to using nonlinear least squared regressions to the output functions, or the predictive quantities that we were predicting, or parameters. So yeah, so that was my general approach, and yeah, it was a lot of work.
Great. Next up is Sergey.
Yeah, hi everyone. So yeah, my name is Sergey. I'm the director of the protein engineering department at Nosumbe Discovery. And together with my colleague, Martin Flor from the Barcelona Supercomputing Center, we mainly also use structure-based modeling tools. So in the in silico part, it was a combination of prediction, like deep learning-based methods to predict of some of the features or labels we had to predict, combined with some structural modeling features. And then the in vitro part was mainly using the structure of the protein and then the information of the mutants to kind of infer what could be the synergistic combination of the mutants that would improve the activity. And this was done with a genetic algorithm. So yeah, this was pretty much our efforts. Yeah.
Great. And next is Han.
Okay, hi, I'm Han. I'm a grad student in Debbie Marx's group. And to echo something that Michael had said right at the beginning, I did not work as a solo person on any part of this. And our lab was like extremely team oriented, especially for the in silico stuff. So most of that was like organized and led by my lab mate, Stephanie, who couldn't be here today because she's preparing to defend her thesis next week. So, and not becoming a father, but, you know, in a way, becoming a mother of a thesis. But a lot of the different things that we used for that round is, I feel like maybe a little bit dissimilar from things I've been hearing from other folks, where we basically tried every single possible thing in our toolbox. So we use some like energy-based models, we use some structure-based models, we use some large language models, we use some small natural sequence models, and just threw everything at the wall. And for some parts of the competition, it made more sense to like take all of our different things and average them. For other parts of the competition, it made more sense to just take certain model scores or just one model score, depending on the context. So it was Stephanie, myself, and then like probably 6 other people in lab, just being like, oh, what are all of our different things we have access to, and just going for it. And then for the design part of it, it was myself and then another person who used to be in our lab named Nathan. And our modeling strategy for that was mostly using family natural sequence models and doing some sampling around that and biasing based on the limited experimental data that we had access to. So in that like DMS and combinations of mutations data, either favoring certain mutations towards things that were shown to be really good there, or biasing away from things that were shown to be really bad. And then trying to design some mini experiments within that larger set of sequences to test.
Great. And next is Hassan.
Hi, everyone. Yeah, I'm Hassan, co-founder and CEO of Medium Bioscience. My background originally is in computer science, machine learning, and then I've been applying it in the area of protein design for the past 4 or 5 years. In terms of team, there were mostly 3 team members that participated. There was me, my co-founder Adil, and one of our engineer, Yasser. So in terms of the approach that we took, so we were mostly participating in the in vitro part, and the approach that we took was mostly actually very greedy in terms of trying to identify the mutations that were positively interacting and improving all properties. So very intentional about removing any mutation that had any of the property decrease, and only work with that pool of good mutation and finding ways to recombine them, either through random recombination or model-guided recombination. And then in terms of the models, yeah, it was mostly regression models purely based on the data. So we actually found one hot to be quite well performing there. And so we used that as a way to score the recombinations. And then at the end, there was MCMC guided sampling based on the regression models to be able to more freely explore mutations instead of constraining the set of mutation that we could try. So yeah.
Thanks, everybody. And just to give context as well for the other two folks on the team, I introduced Chase, who is going to lead the next section, but Pete is also here, who is a member of the Aligned Innovate team and is part of the planning committee. So we'll be filtering your questions. But I think that that's it for the intro. So Chase, please take it away.
Great, thanks, Dana. And thank you again for everyone being here. So as Dana mentioned, I'm going to be going over a set of questions for our panelists. They're going to cover three main topics. The first will be focused on the tournament and your experience there. The second one will be about your experience as protein engineers outside of the tournament. And the last one, we're going to try to see if we can get some hot takes on the field as a whole. And so we'll see how that goes. So for the first question, I would like to ask, as Dana mentioned, the tournament had two rounds. There was the first round where you predict the properties of protein, and then the second where you generate a protein with given properties. So I'd like to understand more about how you think about these two goals. Which do you find to be more technically challenging? Which do you find to be more interesting, either in terms of the technical component or in terms of the applications? And when answering, please feel free to discuss your experience in the tournament. If you only participated in one of those rounds, why did you do so and not the other, and so on. And for this first question, I think we can also do this popcorn style, going through each participant, and then maybe in future questions, it could be more of a grab and go kind of thing with whoever wants to jump in. So yeah, I guess we can start again with Michael.
So I'm probably heavily biased, but for me, the easier part is the property prediction part. I mean, I worked on this a lot. I have to admit, and this also makes the design part a bit more interesting for me personally, because there it's a lot more about being a bit creative. I think for the prediction part, we already have sort of a good understanding of the do's and don'ts. And this is why I'm just more curious about the design part, but this does not mean that I would like to undervalue the importance of the property prediction part, because after all, I need to somehow rank and score the designs, because I can easily generate 1 million proteins and then they are all garbage, so I need to rank them. And this is why I think actually sometimes that the property prediction part is actually the more important one. So you can just let a language model generate an arbitrary number of designs based on some wild type in essentially no time if you have a GPU, and then it's rather a matter of ranking them properly. So this is, yeah. So this would be just my take on this. I'm not sure whether I forgot some part of your question.
No, that's great. Thank you.
Okay.
And I just posted the question as well into the chat, so nobody forgets as they go along. Thank you, Han, for that recommendation. So Ingmar.
Yeah, so I would second Michael that the generation part is for sure the more challenging, just because it builds upon the property prediction. Also because it just takes a while to benchmark this. Just to run this takes much longer than benchmarking property prediction, because you have to be really smart about how to benchmark it. And I mean, we'll talk about it later, but there's a lot of very recent work about benchmarking suggestion algorithms. I think what is different from the approach that Michael is taking to the approach that we have typically been taking is that we build upon known building blocks much more than the Rost Lab did, I think, in the tournament at least. So we are thinking, so I mean, Harry was making everything up on the spot for the tournament, as said, based on also known methods. But typically we look at Bayesian optimization, which heavily is based on a prediction model and then some good theory on top of that. So in that sense, maybe it's a more natural step, but at the same time, we've learned to not underestimate experimentation also because of the tournament, because the creative approach that the Rust Lab took obviously worked. Yeah.
Great, thank you. And Jason.
Yeah, I think probably both are interesting to me. Protein property prediction and protein generation, but yeah, I think they kind of go hand in hand. I mean, yeah, you're generating a protein, you kind of want it to have some specific properties, so you need to be able to predict it accurately. Otherwise, why are you generating it? So you kind of need both. The generation is probably maybe the more fun part. Prediction is more sort of this, more of an exercise for me. But yeah, I think for my case, I was doing like property prediction using structural models, and in some cases, like actually a lot of these cases, the prediction is already established in the literature. So, for example, it's like shape complementarity of the enzyme with the substrate, could lead to higher binding affinity, for example, or a lower delta G of Gibbs free energy binding. Stuff like that, it should lead to higher affinity. So that's already kind of known, and there was already a nice filter in Rosetta that got published, a secretion prediction filter. So that was kind of something I used because one of the properties we had to predict was secretion for one of these events. So it's kind of nice that a lot of this was already available in existing software. So I utilized that. And yeah, I participated in I think all the all the rounds, basically, all the events.
Perfect, thank you. And Sergey.
Yeah, so I mean, I agree with a lot of things they're saying. Like, I think to design proteins, in the end, you need to be able to predict the property of interest or the multiple properties of interest you want to improve on, right? So for us, I mean, I think people tend to think that design is harder, but for us as modelers, for us it's more like, and especially how it was designed on the tournament, like for us it's harder to do the prediction of properties building machine learning models than going on more rational design, classical way of rationally designing enzymes without any data at all. So for us, it was easier, not easy, I mean, of course it's very challenging still, but it's more natural for us to do design than go for predicting properties or the fitness of the enzyme, right? But I think both are needed, and we have to improve on both type of tasks. But probably AI models now are very good at it, and that's why also the teams that use language models or the type of tools probably did the best. Yeah.
Interesting. Yeah, thank you. And Han.
Yeah, nothing incredibly insightful or new to add to the discussion because I feel like most points have been hit. I guess one thing that I want to mention and stress, less so in the design round, more so in the in silico round, is something on the property prediction side that is challenging, is just in general predicting non-natural functions in a completely unsupervised way is like very much not solved. And I find that really interesting because it is hard, and trying to think about if you had some enzyme that works on substrate A and you want to make it work on substrate B without having any labeled data, even if we'd have access to it in the competition, but without having any unlabeled or without having any labeled data, how would you actually move the sequence in that direction? And then just in terms of property prediction, that's challenging. And then adding the next layer of design on top of it and saying, okay, we want to design for this, that's also very challenging. Because then also, as people had mentioned, when you generate 1 million sequences and you only want to test 100, how do you go from 1 million to 100, especially when you have a specific property that you want to optimize? And so it's a balance between, okay, we know we can predict the other things like kind of well for some proteins, which is also a big if it's a protein where there's not many access to natural sequences, you also can't really do that well. If there's a protein where there's no structure solved, you also can't really do that well. Large language models are known to scale their performance based on how many examples of it are in their training data. And so I feel like there's some interesting spots in the protein universe where there's still a lot of challenges. And I find myself, like most projects that I work on in my like regular research is around that. And then the projects related to that in this competition, I also found really interesting in terms of like how to think about the modeling in like a quick way and not like a spending 2 years on a PhD project sort of way.
Very interesting. Thank you. And Hassan.
I think I would like to add to what Anne said here on the zero shot part. I really find it more of a dark art, especially when especially when in the substrate switching type questions where you may just have one model for activity, and then now you have to get into the nuanced understanding of activity on different substrates. So I found that part to be fascinating and interesting, because I think the first principle modeling, it's still an open question on like how to best approach that. So I found that fascinating. And then, I think the generation why a lot of people are excited is that not everybody always has like experimental resource or the frequent cycle to be able to. So it's always like a learning experience when you do all your strategy and you're like, okay, at the end, let's see what was right, what was wrong, and there's always a good surprise. So I found that to be always like a fascinating experience when the result come back. And
Definitely. Great, thank you. Yeah, so I guess some common themes. Everyone, I think, ended up having somewhat of a unique angle, but I suppose some of the common themes were that generation can sometimes be more difficult because you're building on top of design or prediction in some cases. And then also, I think what almost everyone was mentioning was the actual iteration loop. So trying to improve anything requires some kind of iteration and evaluation. And with prediction, at least you can have a data set and do these iterations internally. But with generation, where the proof is in, the wet lab pudding, it just becomes a much more challenging process, which is interesting. I personally think it kind of mirrors some of the large language modeling versus robotics trends that we've seen in the outside of protein engineering world as well, where it's the underlying technologies are similar, but it's the evaluation that really defines your rate of progress. So for the next question, I actually want to focus on that a bit more, is on benchmarking and how you do it today outside of the tournament. So when you decide to develop a new model, and you ideally want to compare it against state of the art, exceed state of the art, so you can have the SOTA claim. How are you choosing the data you're using? How are you choosing the models you're comparing against? When you compare against models, are you often having to retrain them, or do you just use the published metrics? And then, some thoughts between how you do this in silico versus in vitro. For this, I open it up to anyone who wants to jump in, share their thoughts on how they're doing this today.
So then I just start. I mean, I think I never retrained a predictor, to be honest. If it's not easily accessible, like I can just download it and install it locally, run it locally, or if there is no web server, then the model does not exist in my world, to be honest. So it needs to be accessible in some way if you're in academia, because otherwise I do not think that it's a model worth comparing against. And this sounds brutal, I know, but after all, I'm with Han on, like, I do not want to spend on a single project like 2 years of my PhD and retraining some of those and just re-engineering all the stuff that might have been lost in translation in the methods part is painful to impossible, depending on how well the method is documented. So it usually boils down to local installation of existing methods or web servers, and then comparing using some time holdout split. So just taking all the data that was published after the method was published and then redundancy reducing against their training data, for example. And then also, this also gives me the opportunity to add additional metrics, because maybe I missed some metric that the authors did not publish, or they didn't add error bars or something, and then I can just do this by myself. How do I choose SOTA? I mean, for some structural prediction, it's CASP. For function prediction, it might be Kappa. For other properties, you might dig a bit deeper. But yeah, so this is just my research.
Yeah, maybe I can put a plug for a data set, for like a benchmarking data set that our lab put together and published in NeurIPS last year, called Protein Gym, which, for the question of benchmarking, how do we benchmark different protein functions with each other? What we did as a group effort was we compiled over 200 different DMS and like other multi-mutant data sets that exist across like many different protein families, many different organisms, a bunch of clinical data in there also, and indels. And so when our lab is making a new protein model for protein property prediction or design in some way, shape, or form, we benchmark against all of these, and that's how we claim state of the art. And then we also have a leaderboard on this website of different different models that have benchmarked against all these different data sets, and we keep that updated pretty frequently. But the point of benchmarking in the burgeoning fields that exist of how do you benchmark something for which there are no benchmarks is an open question that we think about a lot also. For instance, like if you saw the Evo paper of like, oh, we created a whole genome model for like all prokaryotes. It's like, wow, that's awesome. What do you benchmark on? Like, what do you even do in that? Or there's a project in our lab also thinking about different DNA regulation parts and if we can model those and create models for those. Again, it's like, what do you benchmark on? Even things like RNA structure prediction or just nucleic acid things in general. What do you benchmark on? Post-translational modifications. What do you benchmark on? So just to say, it's a huge, huge, huge open question. And we've put a lot of thought and care into curating this set for proteins, but I feel like it's a massive question in the field of biology and machine learning in general. So great question.
Yeah, one thing.
Oh yeah, please.
Yeah, one thing I would like to add as well. Yeah, definitely good shout out to Protein Gym. It's super helpful for different properties and different scenarios. One other resource I would like to point to as well is something called Flexes, which does a synthetic fitness landscape. So I think one of the common questions when doing strategies for protein design is you want to create the scenario. Okay, I'm gonna use this strategy to pick my original mutant from round one, and then from the round one do the round n. Like tools like Flex allow you to basically build a synthetic fitness landscape where you can have prediction for as many variants. Often those can be model guided prediction, but at least we can get into like scenarios where we look at like 1, 2, or 3 rounds and start to think about what's the difference between random one round, round 2, round 3 versus model guided. You can start to have a sense of like how different strategies work. And you can also use custom data to make those fitness landscape to start doing some of those scenarios. So it helps to think on the multi-round scale.
Yeah, maybe on the note of Protein Gym, I think the one thing that we're lacking, and please correct me if I'm wrong, is a kind of public leaderboard which people can add their models to, like the LLM leaderboards that Hugging Face does, and just get the new benchmark for the new model after, I don't know, 2 hours or something of compute time. I think that's what really would push the field forward further. What Michael said, that's also the case for us. If something's not reproducible, it basically doesn't exist. That's actually a problem coming from academia, as we all do, and then moving into companies. It's like, yeah, okay, it would be better to make something reproducible for everybody, but then where does the unique selling point go?
Interesting. And on the line of data sets like Protein Gym, are people currently benchmarking on individual data sets, training them one at a time to see their performance on them independently? Or is anybody working on this kind of multimodality, if you will, of training across all of the data sets and seeing how it can work across different units and different assay types and so on?
So I would just give it a shot. But so we trained actually a variant effect score predictor by essentially just predicting conservation of residues and then just classifying, irrespective of which protein you're currently looking at, residue positions that are predicted to be conserved as highly to be having an effect on function. And this turned out to work remarkably well also for the Protein Gym datasets then. But we did not zoom into special proteins within the protein gym. So we try to keep it more general, and just it was really as easy as putting an if on conservation. So if conserved, then don't mutate, and then you actually get pretty far with this, surprisingly far.
I see.
Yeah, so like from my side, I think like if you want to model any parameter or like multiple things at once, that's why you always have modeling, right? And you have different levels of accuracy at modeling. You can go from just docking the substrate into a place to doing QMMM simulations, right? So there's a lot of like realms of accuracy, but I guess like people want a model that to be able to predict and then design new proteins fastly. And that's why AI has entered like in the field so drastically. And I think that like a model that could probably do all the tasks like eventually is probably like a protein language model that has enough, like it's so many data, like not just the one that currently is in public data sets, but much more protein sequences that we don't have yet in the data sets, like from metagenomic samples and these things, and probably having enough complete data set that can have rich embeddings to predict any property of interest. Probably because in the end, we always say that everything is encoded in the sequence, right? You wouldn't need the structure and anything. Theoretically, everything is in the sequence. So if we have a good enough evolutionary model and a sequence model that takes all that into account, probably you can predict anything. So that's our view on this.
But then how do you go from the sequence to the function? I mean, that's still, you can have a perfect embedding, but you still need to be able to translate it to the function. I think that's the thing that's missing if we have only unsupervised sequence data. We need to have something that allows us to get a hold of the translation into function, right?
Yeah, I think that's where you need like more experimental data sets, more benchmarks like Protein Gym and make them bigger. And also like maybe combine, like you can accelerate modeling nowadays with like do some MD simulations, and then you can train like a machine learning model that does those simulations much faster than just doing a classical MD, right? And machine learning built force fields and so on and so on. So probably you can like still infer a lot of properties computationally just and not relying on experimental. So I think it's getting more data and then also accelerating the simulations, right? That's, I think it's what will work the best, yeah.
So I would just briefly like to add function definition in itself. I mean, just having tried to predict gene ontology terms once in my life showed me how painful this is. So for EC, it's obvious. I mean, there you just have this nice, clean hierarchy. So for enzymes, this is relatively straightforward. But for gene ontology, I mean, you train something like beginning of this year, and then you go back checking now your predictor, and suddenly the underlying ontology has changed potentially fundamentally. So there is some it is not easy to define functions, so I understand the problems. But I think this is also a major point that would still make the machine learning predictors also better if we had, I'm not even sure which way, but a different and easier way to define function maybe. So just adding to this, not only more data helps, just also how we actually classify, bin, sort this data then at the end of the day to make it also computer readable.
Yeah, the entire topic of how do we actually define function, what are the representations we use, I think is really fascinating, and something I think we haven't spearheaded a huge amount of research on, because just getting the data to begin with is already tricky enough. We kind of just work with the ontologies we're handed. But it would be really interesting in the future if we do end up in a more data-rich world, if we can go beyond some of the ontologies today, which sometimes don't really have intuitive distances between them. When you have like a number and so on, like how do you measure that? And exactly what you're saying, hierarchies as an ontology are kind of, it can be limiting. And so it would be interesting to see more about that. A question I want to ask. So now I want to transition to a larger question on the topic of scaling laws. So I think this is a phrase that's on everyone's mind throughout the world in terms of artificial intelligence more broadly, but I think also in the protein engineering world as well. So what we've seen, as a quick recap of the past few years, these large language models outside of the field of protein engineering, so these generative pre-trained transformers like GPT 3.5 and 4 have been able to achieve incredible success just by scaling up the amount of data that they're ingesting and then the amount of compute through this, chinchilla optimized ratio. And I think there's a pretty massive question of whether or not we're going to see the same thing in the field of biology and the field of protein modeling. So yeah, I just want to open it up to you. What are your thoughts on this? Do you think we're going to see something similar? Or do you think there's something unique that makes us in our domain different?
I think I would start on this. I think the big difference is that what people in natural language do not have is single words, and they need to generate single words. They have full sentences, full texts, which have long range relationships between them. And that's different because what we have is kind of the word level grammar of proteins. So we know how typical proteins look. That translates to knowing how an English word looks. In German, it's really, in German, you can stack really long words together, and that's kind of within word grammar. This is the situation that we're at with protein language models that are based purely on protein sequences. And I think that is the part that's missing, the relationship between the protein and the rest of the world. Biomap and maybe now also evolutionary scale, I haven't read this new paper, they've had some success by just bolting on new types of losses, like making the most out of the data that they have. But still we have the problem of how does the protein relate to the rest of the world? And one thing that, and this brings us back basically to functional data, right? One thing that you can do in enzymes is you can look at the relationship by looking at, well, this enzyme catalyzes the following reactions, and kind of put that information in. And that's something that Noelia Ferrus has been working on. You can put the EC number as a first proxy for that. There's also something that she particularly has been working on. For antibodies, you can look at the binding partners and so on and so forth. But that, I think, is the limiting factor for sure.
Maybe just adding, I think ESM3 wanted to make exactly this point, just scale it up and it's getting better. So this is...
Yeah, I think one of the beautiful aspects of ESM3 is it's reasoning over sequence structure and function, and it's introduced some new sort of invariant geometric attention mechanisms to reason over the structure. And I think that's just like one massive model, and yes, it's pretty amazing, but it still only works with proteins only. I know their example was GFP, but that creates a post-translational modification for the chromophore. It still had some amazing results. So I think once that's going into more of an all-atom type of modality, sort of like Rosetta Fold all-atom, it'll be really, really amazing. And maybe even mixing into these like mixture of experts models. So you can have these smaller models and sort of that it can take tokens from or results from. That I think, it's just an amazing amount of progress so quickly in the field that I think scaling is a big part of it, but also the methods will really help.
Yeah, I'm happy to comment as someone that came from natural language processing. I think to state the obvious, in natural language processing, the scaling has been like more successful in the sense that the 100 billion parameter model is like way better on almost all dimensions versus a 1 billion. Although people are now getting smarter about the data quality that they're using, but we've definitely see those step jump like between BERT and GPT and Claude and whatnot. And I think like in the protein space, that debate, is still open. I think there was a lot of work, even included by Michael and others, and the Mark's lab, that there wasn't initially as much gain in scaling. Like you could have like some billion parameter model that were as good as 100 billion, or at least you would not see that level of difference. And like that was a bit of the consensus. You know, there was a lot of papers like scaling the limit of ProGen. There was REDA. There was like a lot of things where like, hey, you know, we tried something at 100 million, we tried something at 10 billion. We just didn't see that level of gains. I think and so one of the solution in the kind of AlphaFold 2 and 3 world was just, we're just gonna have better model that harvests what we have, right? Just really good model on like a few 100,000 PDB structures, and like that's gonna get us really far. And so going more in the kind of physics-based modeling world, or at least like geometric aware, and that has been quite helpful. I think, yeah, ESM3, like it kind of comes with like, you know, interesting learnings. I mean, I think that it's still TBD because there are still some tasks, some specialized tasks where like much smaller model that have more complicated prior still outperforming. So I think it does put a few things into question. I think if I were to bet, I would probably still go on like more complicated architecture that take more advantage of the data that we have. And like I think that there's still a lot of room there to do better simulations of the physics that's going on, of the binding that's going on. So I would bet a bit more heavily on that, but I do agree that yeah, you know, recent advances have maybe put some shown that like we've not fully explored what scaling means, and maybe we're not fully tapping that. But yeah.
Yeah, I feel very similarly to that. And I feel like it kind of goes back to the benchmarking questions also of like, okay, if you have this set of benchmarks that's really diverse, and on average, ESM3, for instance, performs really well. But if you're a protein engineer in a lab who needs to design a new enzyme, is that the model you're going to use? Maybe. Me? Probably not. Like I'm going to use like what Hassan had just said of like more complicated architecture, more focused search for some bespoke task. And so part of me also wonders like, yes, it's important to track how these things perform relative to one another in general, but something that we do in our lab is also break them into different tasks of which data sets fit into which buckets. And for instance, a binding data set is going to look really different than a stability data set. And in some cases, we'll want to know generally what performs better overall. But in other cases, we're going to want to know what's actually the best stability predictor. And so whether that's focused on one data set, one protein, or a collection of data sets where that's really the task behind it, I feel like that also is really important to keep in mind. And in terms of scaling, yeah, something that I don't think has been mentioned yet in the discussion is also this idea of the unknown, which of course large language models and things interact with also, where you say like, come up with a new idea about this. And it's like, oh, okay. But I feel like for us also as protein engineers, we want new to nature functions and we want things that have never been seen before. And so I feel like that is something where it's a little bit hard to know whether scaling will ever get to that. Even if we had every single sequence of every single thing possible on this earth and every single structure of every single thing, there's still going to be stuff that we want to do that has not yet been seen. And so how do we actually do that well is an open question.
Yeah, so we have like related to the scaling up of the language models for proteins. I mean, we have recently seen in our team, we published in the ICML like that whether protein language models are compute optimal, right? And we saw that regardless of the model size, increasing the model size of more parameters, you didn't improve the performance. It reaches a plateau, right? And that's because again we are not exploring a space of the protein. Like I mean, of course, the protein sequence space is immense, but we are not exploring yet all the protein sequence space that can be probably expressed and have function and so on. So we are lacking a part, like it's like if we were lacking some part of the English language, right? So that's why probably the protein language models are still not fully not predicting all the properties very accurately as well. I mean, they are quite good, but we have to improve because there's some rules of the protein universe that are not there because the data sets are not yet full. So that's one way of we like how we see it for the like. I think that is the thing that will get closer to a general model But yeah, I think that also that's true that the new to nature functions maybe will need more focused models rather than just a protein language model that uses Earth protein sequences. Yeah, that's true.
Yeah, those are really interesting points. It is such a large unknown between this idea of a generalized model that can do everything, that can just continue scaling and be able to generalize across any new function that we want, versus these very specific bespoke models that have perhaps really strong priors and really curated data sets around their function. It's a large unknown of what the paradigm might look like 2 years from now, 5 years from now, 10 years from now. It would be exciting to see. And so another question, so a lot of what we've been talking about, especially with the scaling laws, focus on data-driven models that have some component of machine learning baked within them. But one question from the audience, from Moimir, was where you see the future of non-data-driven models. So I think more on the terms of first principle, simulation-based, physics-based models, which once were the dominant form in protein engineering. I know energy and excitement has shifted, but where do you see those playing a role now and perhaps in the future?
Well, I'll just say, I mean, I'm not an oracle here on this, but I think probably like to accurately predict the sort of quantum chemistry we'll need more or less erroneous, I guess, error-prone quantum computing hardware. And once that's available, and software, I mean, in terms of the software, that's compatible with the hardware. But computing, the quantum chemistry is extremely challenging. So if there were machine learning models or AI models that could estimate those functions, then we would probably get pretty close to having like a generalized model.
Yeah, I think that's like what I said also before. Like, I think we have to also add machine learning models for accelerating molecular modeling in the end, and avoid like the high computational cost of doing a molecular dynamics or a quantum mechanics calculation. Like, I think that's where the more physical based models will improve in the near future. In the present, I don't see coming up with a new laws of physics equation. Like, I think this has been solved for a while. So I think it's coming up with smarter models, how to sample more smartly, so enhanced sampling methods, and I think that's it for modeling.
Okay, and so thank you. And so the quantum computing, it's always been something that's just there in the background, and maybe it just one day appears and then it simulates everything and we don't need machine learning anymore. But it's also like fusion, where it's always 30 years away, so it's hard to rely on it. So for the final question, I just want to end with some key takeaways from the tournament. Anything that you learned that really changed the way you see the field, might change your research going forward. And it could be specific details about another participant's method as well, as something, just any tangible insight you benefited from.
Yeah, so for me, for the special in vitro part, like, it was that I thought that modeling would go better for thinking of mutants that act synergistically, and sequence-based models or like language models or other things would be more giving you mutants that make sense, but not a super active mutant. And I was surprised that it was the other way around, kind of, at least for our case. So that was a good lesson for us. Yeah.
So for me, it just showed again the importance of good ranking, good property prediction. So this was for me just like, generation design is fancy, it's amazing, it's a lot more fun, at least for me, but the other part is so important, so better not jump too quickly over it.
Yeah, something that Nathan and I, the other person who did the designs with me, has been thinking about a lot lately is how to balance the risk of trusting experimental data outliers. So the Ross lab, for instance, took the #1 performing multi-mutant that we had access to the data and basically did a bunch of things on top of that. And from us, our perspective was, ooh, that's really risky. We don't really want to put all of our eggs in one basket, especially because that thing looks like an outlier. Like it's not in distribution of the rest of the stuff. And so it seems like maybe there was a weird measurement or maybe something else. But I feel like something that was fun to think about and look at in retrospect was being like, wow, yeah, you like in this time, maybe it was luck, maybe it was just like really good experimental measurements, but like that's awesome that they were able to do that and not have that thing break everything else. And so I feel like that is another learning and thinking about how do you prioritize the diversity of your starting point such that in case you have all your eggs in one basket, it either turns out really well or really horribly. And how to like balance that. And I feel like we got a good sense, a good piece of inspiration for future projects of like, okay, cool. Maybe that is like a really good approach.
Perfect. Okay. Excellent. Well, sorry, Jason, do you want to jump in?
Oh, very quickly, I would say, yeah, I also learned a lot from everyone else's methods. So, and thank you for sharing that all. And yeah, the Ross lab in particular, I learned you should take advantage of existing latent spaces that for polysaccharide hydrolysis. That's something that I kind of wish I baked into my models as well, because there's, yeah, just a lot of data out there, and our in vitro challenge was to optimize something that had a lot of data. So kind of taking advantage of that latent space was a great idea.
Perfect. Okay. Well, thank you everyone very much. I know we're coming up at time now. For the participants in the audience as well who submitted questions, we appreciate that a lot as well. I know we weren't able to get to all of them, but please, I know one of them was focused on the experimental side and the in vitro validation in-house. And that's the kind of thing that we would love to answer if you wanted to follow up with us directly or message us on on the Slack and so on. And we'll also be, as Dana mentioned, publishing a follow-up paper that includes more details on the pilot tournament itself. And we're going to be touching more on what we think about the experimental side as well, because that's a huge component of this entire ecosystem. And we have talked about the importance of having better benchmarking and having better access to models, right? The kind of stuff that Hugging Space has for model hosting that makes it easier to compare. And one big component of the tournament that we discussed as well is having the same kind of reproducibility and accessibility on the experimental side. And so there's various technologies and so on that we want to explore and incorporate to the tournament to make the science itself more open and the reproduction of the experiments more open and accessible. And, we talked about that a bit in some of our previous presentations as well. But yeah, I guess I'll hand off to Dana if you have any closing words that you'd like to say.
Yeah, as promised, we have this little QR code for those folks who want to check out our website. It's also going to alignbio.org and looking at our tournament pages. But we have, a lot of different ways to get involved. You can participate in the next tournament. Our goal as of now is to host the next official tournament in 2025. And on our website, you can go and sign up under the tournament pages to be on that mailing list so that you can be contacted immediately as soon as we know what that's going to look like. We are moving forward, hopefully next year, with creating data sets that are specified by the community and our tournament committee to investigate maybe possible new tracks, different enzymes, maybe binding. And so we're going to be creating data sets, but also we found out that through this year, through the pilot tournament, that the tournament is a great way for people who have privately held or unpublished data sets or data sets that are behind patents to be able to donate those data sets to direct people in the field to work on their area of interest. So if you have a data set that you'd like to donate to the next tournament, please feel free to contact us. And then if you'd like to be a part of all of this and have more of these kinds of conversations with these great teams and decide what the next direction of the tournament should be, we're looking for people to expand our planning committee and be a part of everything. So I think with that, we're going to wrap up. Thank you everyone who attended, and thank you again to all of the teams for doing this. Again, it's been so fun to hear you guys speak. This is being recorded, so we'll be getting it, editing it, and putting it on our website. So feel free, we'll send out the link, and feel free to share this amongst the community. And like Michael said, please go forth and spread the message. The in silico round, we can have as many people as we want join and participate and be a part of it. So please let everybody who you think you want to vie against in this space now, so that you can see how their models versus yours look. But with that, we're 3 minutes over. Sorry about that. This went by so fast, but I'll end the recording now. And thank you again, everybody, and good job, I guess, to all of the teams in the tournament.
Oh, can I say one thing, Dana?
Oh yeah, I'll keep recording.
Okay, one very short thing, just to say thank you to everyone on the Align team and everyone who planned this, because honestly, enormous amount of effort, very well executed. And I feel like I can speak for myself and every other team that we all deeply appreciate it. So thank you for everything that you do, Dana, and Pete and Chase, Hassan, everyone else who was part of the planning committee, because truly an enormous effort that you all orchestrated very well. And I think Mark's lab and other people are really excited about future competition. So thank you so much.
Thank you for that.